Is it good now?
Yes.
Yeah.
So it is a pleasure for us to have Michael Herzog with us. Michael studied mathematics, biology, and philosophy at the universities of Erlangen, Tübingen, and in Massachusetts, MIT, at the US. He has a PhD in biology. He did his postdoc at Christoph Koch's lab at Caltech in California. And he's been a senior researcher at the University of Bremen, and since 2004, he's a professor for psychophysics at the Brain Mind Institute at EPFL in Lausanne, Switzerland. And we are very glad and delighted and honored to have you here and to be virtually in Switzerland. And welcome to our seminar.
Okay, thank you very much for this kind introduction. Thank you very much for inviting me, and it's a great pleasure to be in Israel, even if it is only virtually. I like to go to Israel because of the good food, but okay, so we can't have this. I hope my connection will stay stable. If not, you can interrupt me right away. And if there are questions, I think I would be very happy to answer them actually during the talk. So I think it's much easier to communicate in an online fashion rather than offline later on. Okay, the talk is aimed to be provocative. I hope this is going to work out. The idea is in some way to really ask the question to what extent the last 50 years of vision research have really gained us something. And for this reason, I just would like to quickly sketch what I believe is more or less not common sensus, but I think the mainstream idea in visual processing. And usually it starts with an image like this. And intuitively, we have the feeling that we see everything in this image, that we have access to all of the elements. But we know that this is not true. For example, if I ask you to look at the guy which has a pink t-shirt and a blue jeans, I think it will take you a little while to find him. Now you can try a little bit. Yeah, it's here actually. And he's here. And it's obvious that we don't have access to the entire world. And we know this from the visual search literature quite well. So for this reason, what most people believe is that actually vision is not immediate. We need to analyze the visual scene. And usually this starts with neurons with small receptive field, which cover the entire part of the visual field. And at each little of these circles, these confined regions, we basically analyze some basic features like motion, contrast, and color. We have circuits for this, and they do a good job in some way. And often actually we relate the output of these circuits directly with the percept. So there is in some way a one-to-one correspondence between the psychometric and the neurometric function. And the important thing is that this is locally confined. So a classic example is surround suppression. Here the task is to basically detect the Gabor patch or the orientation. And here we have spikes per second. Can you see my cursor, the mouse?
Yes.
Yeah, good. Yeah. So you can have here spikes per second, so that's neurons that fire to some extent. And you have this also psychophysically, here you can just plot performance. You get a very similar plot. Then you increase the size, and not very surprisingly, there are more spikes per second. And also in the behavioral experiment, you get a better performance. And then the surprising thing happens, if you increase the patch a little bit further, then the performance and also the spike rate goes down. And that's one of these many classical phenomena from psychophysics and neurophysiology. And what we do then next is basically, I think, spend many, many years on finding out what is the correct underlying circuitry. And then there's these questions, is there inhibition, say, at V1, or is it from extra striate areas, or actually within the LGN? And I think we spent many, many years understanding these basic circuits. And the key idea behind this is that these basic circuits are something, yeah? Oh, okay. That these basic circuits are an alphabet of perception in some way. So if we understand what's going on in these locally confined environments exactly, then we know basically what's going on at the basic stages of vision. And then what we can do is next, we basically combine these outputs from these many neurons with small receptive fields. And so in some way, we can perceive more and more complex things like this visual scene. I think this is something most people believe, and that's part of this standard feed-forward processing scheme where we go from the analysis of small elements like edges and lines in V1 to shapes, objects, and faces. And a cartoon is a little bit here. So we have this rectangle here. Here we have our small neurons. They code for orientation. We have a bank of filters at each location with all the orientations. We have this for the next spatial position and so on and so forth. And then what we do is we combine this. For example, here we have these line detectors, and then we can combine line detectors with a logical end if you want, and we get some corner detectors. So here, for example, you can combine this with this. And at the end, you combine the corners and you end up with a rectangle. So in some way, we have this pooling of information from small receptive fields to neurons with much larger receptive fields that become then object detectors. That's a little bit the idea. So we go from low level to high level processing. And in some way, the low level processing strongly or fully determines the high level processing. I know it's a cartoon, but I think this is behind many, many models. And for example, the classic CNNs, classic deep networks, they work exactly in this way. Okay, here's another example, and that will be the example we might use during the talk, or we will use during the talk. And I think you're familiar with it. If not, you can experience this yourself. So you can stare at this bull's eye here, and you don't move your eyes. And now your task is to look for a kid. Don't move your eyes. Can you see the kid? So you can move your eyes now. And you see here's a kid. That's easy to see, but here's another one. And that's hard to see. And the reason it is crowded, that's a normal situation in life. And the strong crowding here occurs basically by these construction signs, and mainly because they share the same color as the t-shirt of the kid. It tells you a little bit how you should dress your kids when you bring them to school. Okay, that's a classic crowding situation. It means that if you have a target, it becomes harder to perceive when it is embedded in clutter. And here's another example. You can stare here at this red line, and you can easily see the letter R. Now I add two letters, and even, you know, it is an R. No, it's very hard to identify. You still see there is something. It's not masked, it's not invisible, but the features are very hard to identify. That's called crowding. And as I said, this is the standard situation in vision because we encounter targets rarely in isolation. It's always something around. And the explanation is straightforward. It is nothing else, as in some way crowding is thought to be nothing else. It's just an outcome of the classic object recognition mechanism. So here we have the letter V, not an R, it's a V now. And the idea is it is basically nicely analyzed by some input, some line detectors, maybe some letter. And then we add some letters, and they're all nicely analyzed on the early stages with small receptive fields. But as we said before, to perceive a rectangle or objects, we need to pool the information. And at this stage, an input gets lost. That's classic situation. In some way, crowding is an unavoidable consequence of object recognition. And the last thing here is, and now research has spent on this for almost 100 years, and the characteristics are worked out quite well. For example, it depends, of course, on the eccentricity. It depends on the distance. And here's a demonstration. You can look here. The middle letter V is hard to see. We move this, yeah, and all of a sudden it becomes visible to you. And the explanation is quite straightforward. You basically move the flankers out of this pooling zone, which is often called Bauma's region. And with this, basically, the element becomes visible because there is no pooling or combination of elements. And this can even give a mathematical expression. And usually this Bouma region is thought to be basically depending on the eccentricity here. and it's just a factor of half. So in some way, this is research, it's good research that has shown how things work out, that has analyzed the characteristics, but it has never ever used more than 2 flankers. So not never ever is not true, but almost never used more complex stimulus generations. Okay, so that's the first part of the talk. I think this is just maybe building up the strawman I want to kill. The idea is in some way that processing is local at the early stages. We can relate basic circuits to basic percepts. The high-level processing is fully determined by the low-level processing, and that features that are lost at early stages are irretrievable builders. That's actually the idea about the crowding. And in some way, the system cannot do better. So in some way, I think it's a nice engineering approach because it tries to break down the complexity of vision. So you have a visual scene. We know we cannot perceive it right away. We need to break it down into its component, and then we need to combine it. I think this is a very good approach. And if it would work, I think it would be very nice. But I like to show you in the following that none of these ideas is actually true. Let me see. Okay, here's our first experiment. It was done by Mauro Manassi, and it's a rectangle discrimination task. And you potentially ever heard about this. It's also not very important. So subjects were asked to tell us if the x-axis of the rectangle is longer than the y-axis. So here you say it's wider, and here you say it's more narrow. Okay, we did this task in the periphery at 9 degrees. We computed the threshold. So that's basically the smallest ratio between the two, or the distance here you basically can perceive. And people need, you can see it's 300 arcsecond to basically, that needs to be 300 arcsecond wider than this line to perceive it. For the threshold, up means bad performance and low values means good performance, simply because you need smaller differences to perceive it. And we run a staircase procedure on this. So that's extremely standard, nothing special here. Then, oops, we added a couple of squares to the left and right, so it's classic crowding, and exactly what happens is you find these huge crowding effects. So there's nothing special, and target is perceived, the performance on the target deteriorates when we add the flankers. Okay, classic crowding.
Sorry, Michael, is it possible, can I ask a question about the previous slide?
Yeah, go ahead. Which one, this one?
Yeah, this one. No, the next one. I wanted to ask what do you mean when there is, when you say that you need a bigger threshold? Do you have this as spacing between the elements, or do you make the actual square rectangle bigger?
Yeah, what we do is we basically have a rectangle here, and we keep the y-axis constant, and we change the length of the x-axis. And basically we looking for,
yeah, I understand. You need to make it wider, much wider, in order to perceive it when there are additional elements. Okay.
Right, yeah. So we make it longer here as the y-axis. So this is always fixed, and it could be wider or smaller, and we look basically for the difference. So different, from the y-axis, and it needs to be 300 arcsecond wider. So then you get 75% correct.
Okay, thanks.
Yeah, fine. No, ask question, please. I know usually, when with a real audience, usually it becomes quite clear if people don't understand what's going on, but it's very hard right now without any feedback. Okay, so this just tells us basically, it doesn't really matter exactly what's going on. You can also use percent correct, but we use the thresholds because of the flow and ceiling effect. So it just tells you you're quite good with one square or rectangle, but if you have many of them, then you get worse. This is really nothing of interest. It's a classic crowding effect you observe all the time. Okay, here's another one. We have your Vernier task. I think you might be familiar with this. So here the participant need to tell whether the lower bar is offset to the left or right. So here is left, here's right. And they push a button accordingly. We also presented this at 9 degrees of eccentricity. And here's the results. And subjects need 100 arc seconds, basically difference between an offset of 100 arc seconds, so you obtain 75% correct responses. Nothing special about this. Then we added a square around this, and you can see again this huge crowding effect. So it's really large psychophysical effect. So it's almost a factor of 6 or something like this of deterioration in performance. So that's another classic crowding effect. Nothing interesting here. And then we just combined the two. So the task right now was on the Vernier, not on the square. So you have to say if this is left or right. And this is more or less, we should now be able to predict more or less the performance by the performance on the two subtasks. So in some way, what we would expect, or what would you expect usually? Usually I ask the audience, but it doesn't work with Zoom. Usually I think what people would imagine, it gets worse because, you know, we have the crowding of the square on the Vernier, and on top of this, the crowding of the other squares on the central square. So we have 2 times crowding of this single target and crowding of the many targets. And of course, you can imagine what's going on. Just the opposite. So here we have performance. So this is good. This square is bad. I add many squares. And you see that performance almost approaches the condition where there is no square at all, where there is no crowding. Yeah, importantly, the square here is the same square as here. So in some way, we don't get more crowding, we get less crowding. So in some way, this is very hard to explain with classic models because you would expect that you have more noise, you have more whatever inhibition or lateral inhibition, and you should deteriorate performance, but it doesn't happen at all. So that's very hard to explain because what we believe is what happened here is that it's not the low-level interactions that determine the high-level ones. It is the opposite. The high-level figural interactions determine the crowding. And that this could be true can be seen with a simple experiment here. So what we did is we kept all luminance and low-level features the same except for the orientation. And, oops, sorry, I forgot something. So the idea is, let me go, sorry. So what we believe is we have these shape interactions first. So no information is lost. And then you basically group all the elements, the squares, and then basically the Vernier is not grouped with the squares and is processed independently. And at the very end, I will show you very quickly some modeling we do with very complex models, which basically give evidence for this. Here's the psychophysical evidence. So what we did is we basically just turned these squares, made them diamonds. And what you can see is then performance strongly deteriorates. Here actually you get a small, even extra deterioration to the single square condition. Okay, what is more important, oh no, that's quite important, but what is as important is, here you have your Vernier, it's bad. You have one square, sorry. You have one Vernier, it's good. You have a square, that's bad. You add 2 squares, it gets a little bit worse. You add 2 times 2 squares, it gets a little bit better. And finally, the more elements you add, the better the performance is. So,
question, can I ask a question? Just to make sure I am not missing something. All this was done in peripheral vision. It's not in foveal vision.
No, it's all at 9 degrees.
Yes, so basically we are talking here only about phenomenology of peripheral vision. If you have done this experiment in foveal vision, will you get the same effects?
Yes.
Okay.
We don't have these results with the squares in the fovea because they're too big. But if you, I have some experiments I can show you later on. If you do similar experiments, we have hundreds of these experiments. This is just the tip of the iceberg. If you do the experiments with very similar experiments with Verniers and lines, you get exactly the same results in the fovea. Of course, you need to scale down the stimulus. So what I believe is it is a really universal visual processing scheme. It's something you find all over. It's not something, a peculiarity of the peripheral visual system. So if you don't like it, you cannot say it's peripheral. But did that answer your question? Okay.
Sorry,
Cool.
I was mute. Very well. It's a good answer.
Thank you very much. Okay, so this is a gradual effect. And actually, if we have not only a line of squares, but an entire grid, 35 squares, you get actually exactly the same performance as just with the Vernier. So the crowding goes completely away. What is important is that the fixation dot is here. So actually that answers your question in some way. The observer's fixating here, and the entire area here, that's basically 20 degrees. So the Vernier offset here is 100, or 200 in this case, and the entire configuration matters. And if you compute it, it's 72,000 arc seconds compared to the 100. So to understand what's going on here in a very small confined region with small offsets in the range of, say, 100 to 200 arc seconds, you need to know what's going on in a region that basically covers 72,000 arc seconds. So it's a huge, huge difference. So what I want to say is what's going on here at one point in the visual field depends on more or less what's going on in the entire visual field, or at least in the entire right visual field in this case. So we cannot know about this point if we don't know about all the other points in the visual field. So that's very hard to understand with this classic idea that first we should basically find out about the basic processing and then we just can combine it. So it seems to be that we need to understand the full visual scene before we can understand what's going on at one point. And that's basically just the opposite of this actually quite nice, but unfortunately not tenable classic approach where we try to break down complexity by analyzing the sub-processes. Which I'm a trained mathematician, so I would have loved to be it this way, but it is simply not true. Yeah, that I said, right? Okay, let me play a game with you. Let's see if this works. Usually it works quite nice with in real time. So here we have our square and bad performance. So Vernier is here, bad performance, good performance here. Now I replace some of the squares by stars. And I can ask you can ask yourself right now, what's going on? Will performance deteriorate or improve?
Intermediate.
Intermediate, okay, that's good. Oops, no, it's actually as bad as one square. Okay, next one. So we basically just double the information. We just copy paste the central line and add it on top and below. You can ask yourself, does it improve or further improve or get better or intermediate?
Of course it will improve because it has to be interesting. I mean.
Yeah. Okay, I should replace these ones. Yeah, no, basically, you have this.
It's a striking effect, I agree.
Yeah, yeah. It's quite surprising in some way. So you have the same row here, you just copy it and you get good in some way. Okay, one more. We just move it by one inter-element spacing. Good or bad?
Bad.
Yeah, it's very bad. But my point is usually, when we do this with a in-person audience, usually the responses are quite mixed. Some believe it's better, some it's worse, and so on and so forth. The point is you don't really know. You cannot predict. And that's actually the main point I want to make here. We're having huge differences, yeah, there's big effects, not small ones, super large effects. And we cannot predict. So, what I can do right now, I can shuffle some of the squares. Actually, there's 30 million combinations. And the point we simply don't know. So in some way to understand what's going on at one point here, you need to know what's going on across the entire visual field, and you need to know the exact spatial configuration. And that's the problem. Yeah, on a local level, everything is the same here. We always have the square. Even at some configuration, we have the same central row, but this is not predictive at all.
Would it be predictive in some way? I mean, I don't know how you could measure some attention-related measures. I mean, I'm thinking that maybe these, you said something about patterns and the fact that maybe it forms a pattern that you can then extract in some way, or the system extracts to put it as a background. But I'm thinking that maybe it has to also do with the fact that in some selective attention mechanism is able to extract it, and maybe in other patterns it cannot, and maybe you can have some attention related measure here that may assist you in determining that, if the focus of attention is localized or spread or contaminated. I don't know.
No, I completely agree with you. We actually believe, or actually we have a paper on this, where we basically show that basically there is a strong link to actually the attentional literature. And the point is, you basically get the uncrowding always when you basically get a pop-out. So if you have a pop-out performance, it's always good. The opposite is not true. So indeed, what we believe is that one of the issues is whether or not you can attend, you can access the target stimulus. That's the point. If this happens, if attention is diffuse, we're testing right now, it looks like it's more a pre-attentive mechanism otherwise. But the big question is, when do you get pop-out in these displays? That's basically the same question. So in some way, you're completely right. It is basically attention, or if I can access the target. But the big question is, what configuration allow me to access this? Or what configurations make this here pop out and which configurations don't? And that's, we don't know. Okay, yeah. So we think that the shape processing across the entire visual field determines the Vernier acuity. Okay, and just quickly, we had Bauma's law, which says it should, all these interactions should occur only in a window of 4.5 degrees, but you know, we saw this is much, much larger. Okay, you might say, oh, these Verniers, what these guys do in Switzerland, that might be fine, but it may be just with Verniers and some specific stimuli, but I think this is not true. We had Gabor's, this is actually also in the fovea here. It basically shows the same pattern. We had letters, it shows exactly the same pattern. We had other not crowding paradigms. We tried it in backward masking, pattern masking, it's all over. Other groups have similar stuff with textures, with noise. Some Israelis did it with patterns. Then we have, this is actually old, 79, they found similar things, Porter. So it's all over the place with almost everything you're trying. And I'm sure if you try your displays, for example, Uri's lateral inhibition, I think you will find similar effects. It is just that we haven't found these effects because we've only focused on flankers, on very few flankers. And you might say, oh, maybe this is something in vision. No, it happens in haptics too. Exactly the same. People are blindfolded, move their fingers over some areas, and they get exactly the same effects. And finally, in audition. So it's really all over the place, and something we need to take into account. Okay, give me a second. Okay, so I need to hurry up a little bit, sorry. Oops, what's now? Uh-oh.
It is 12:30.
Yeah.
Yeah.
Sorry, do you see a black screen right now?
We see the main screen and the slide sorted on the left.
Okay, good. Do you see dynamics of vision?
Yeah.
Okay, good. I need to hurry a little bit. Now we come to something completely different for a minute, and then we get back and put everything together. So this is a demonstration that doesn't work. I tested this with Sharon before. What you should see is basically a yellow disk, and it is not yellow. It is basically a green disk, a red disk, a green disk, and a red disk at the same location. All are presented for, say, 20 milliseconds. And you cannot tell if there is a red or a green disk or the order. You just see a yellow disk.
I just want to clarify that we don't see it because of the zoom temporal resolution, not because it doesn't work.
Yeah, thanks. Exactly. And here is basically once more, here is just with two disks, red and green gives yellow. And we can do this with the Verniers too. This is right, this is left. They're presented for 30 milliseconds. And as with the colors, they fuse. You cannot see that there are two Verniers. and the offset basically is just straight. Always the later element determines the percept a little bit. So here we get a yellow with greenish tone, and here Vernier that looks a little bit more to the left. Okay, we asked subject, tell us it's the first, forgot to tell you something. And what we do is we ask the subjects about the Verniers, and if this goes to the right, this goes to the left. And if this goes to the left, this to the right. So there's only two stimulus combinations. They never go in the same direction. And we asked them, is the first or second one offset to the right? And people cannot do it. And you would have believed this right away if the animation had worked. And then we do TMS. This is quite fun. This is 1911. That's how we do it. And it more or less does the same thing as before. And it's just a little bit more convenient. And what we do is right now, we basically first determine the performance that in terms of the first Vernier. So you see only, as a participant see only one Vernier. They cannot tell apart the two. And here they, for example, would say it's offset to the left. And what we plot is the accordance with the first Vernier. So this was in accordance with the second Vernier. So if you get 100%, here we are, sorry, in the task before we ask, is the first or the second Vernier offset? And you cannot do it, so it's unconscious. Here we ask, is the perceived fused offset left or right? So actually they didn't even know about the two Verniers in this experiment. And what we plot is we plot performance in accordance with the first one. So here if you say it's offset to the right, always offset to the right, then you get 100%. And if it is offset to the left, is in accordance with the second, you get 0. Yeah, that can be left and right, but if it is in accordance with this one, you get 0, here at 100%, and at 50%, it means both contribute to the percept. Okay, and then we zoomed ZEP TMS. So here again is our stimulus, right, left, seen here again, it's 60 milliseconds. And after 60 milliseconds, nothing is on screen. Here is again the performance for when we have no TMS. And here we plot basically the TMS when the pulse is elicited, when we zap it. It's a between primal visual cortex and V2. And now assume, for example, the TMS has no effect. Yeah, I put the coil in the air or the field is too weak. Then we would expect that this curve is flat because it doesn't matter when I zap TMS. If it has no effect, it has no effect. Yeah, so it should be basically on top of this dashed line here. Now assume that the two Verniers fuse right away. So basically the two Verniers integrate immediately. It's like a drop of milk in coffee. Then again, we would basically get a flat function. Because by definition, that's the trick of the experiment a little bit, the 50% dominance level is the same as chance level. And TMS cannot basically get the offset back. But to the contrary, so if this curve is not a flat function, it means that the two Vernier offset are not fused yet. So there, in some way, there must be representation of each one individually in the brain that are active, encoding for this specific one, not for the fused one. That's the idea. And if you look at this right now, we can see that between, say, 0 and 150, actually the second Vernier dominates, which is a little bit paradox. We have no explanation. And from 150 milliseconds on, actually the first one dominates. Yeah, you can see this here, for example, the effects are quite nice in some way. So this goes down to 35% and up to 68 or something like this. So if I zap TMS 180 milliseconds, you're more inclined to push the button in accordance with the first one. And this goes up to 420 milliseconds. At 360, we still have a significant effect. So that means even though they are presented for a very short time, yeah, after 60 milliseconds, show is over, still 300 milliseconds later, we have still an effect. So the information gets into the brain and it stays there for quite a while independently. And we believe that's the point. We believe that there is a window of integration which combines all the information. It stores like a movie in some way. And after the end, basically, the information is collapsed into the conscious person. And here we have some controls. Basically, once more, we can show it. The TMS doesn't work on the feature detectors. This is just this experiment. It cannot work by definition on the fused Vernier. So it means we have this feature memory, we have some representations that are independently stored. And then the TMS basically, or this processing of the two is vulnerable to TMS. It's like two kids on a seesaw, and the TMS basically puts one over the other one. So we believe that the consciousness does not occur before 420 milliseconds. It's discrete here. We are only conscious at certain moments of time. And I skip this. So that might sound a little bit strange, and the question is what does it has to do with
Michael, just
yeah.
a question.
Sure.
Actually, the difference between the two is only the one line, the upper line, either going to the right or to the left. The lower line is the same. So it's kind, you have two different detectors, one detecting the right one, one detecting the left one.
No, actually,
so they have competition between them.
Yeah, that's the point. That's the point. Actually, the verniers on the screen, this goes really, the midpoint is here, this goes a little bit to the left and right, and here it's the opposite. Does it answer your question, Ulrich?
Yeah, but what I'm meaning that the one possibility that you have the changes that you have two different detectors. One is let's respond first and they inhibit the other, and then they can switch.
Yeah, that's what we believe in some way. In some way, what we believe, you have these memories. I'm not saying they are static memories. What I'm believing is, and we will show this in a minute, what I'm believing is you have these detectors that interact with each other during this long period. So the information comes in, the detectors are activated, but you don't have an immediate percept. You have quite some time where these elements now start to work with each other. And the message layer, which I cannot prove, but my feeling is that even for static stimuli, like the crowding stimuli, you have exactly the same thing going on. The information comes in, it is buffered, and in this buffer, you start basically doing all these complicated visual processes basically to solve the ill-posed problems of vision and to come up with a meaningful percept. Okay? Okay, and this sounds a little bit strange in some way. And the point is, I think we can show that this makes sense. As I said before, what I believe is information comes in, and even if it is shortly presented, like with a TMS, it takes a long time until it is processed. And I think the same is true in the crowding. The same is true in general. What we do is we basically store an entire movie of half second, and then we collapse the information. And here's another demonstration for this. It's too bad that I cannot show it to you, but there is a demo. If you go to this paper, you can find a demo in animation. That's at least what I hope. But I'm going to show you here is basically, you see, I hope you can see my fingers right now. You see a stream of lines, something like this. Yeah, it looks something like this. It's two lines that expand from the center. So that's seen here. That's how the stimulus looks like as we present it. And in one frame, we have a line, and in one presentation, we have a line, and in the other, we don't. We ask the subject, is it this one or this one? And I could show it to you, but you cannot see it. You would believe it right away, because if I show this two to you, you would always say, say 95% say this was this presentation, but in fact, it was this one. So what happens here is because of this metacontrast type of masking, we remove this element from consciousness. So it is there, but you can't see it. Yeah, it's here. So they're at 55%. The surprising thing right now is if this line, which is not visible to you, gets in vernier offset, so we make it offset to the left or right, but all these lines are straight. You now perceive all the lines, or this moving one line, in the same direction as the vernier. So if this goes to the right, this goes to the right. The entire stream goes to the right. Even the element itself is visible. The offset, its feature, basically is perceived at the other elements. And we can measure this. So here we have attention here. Performance is quite good. Subjects don't even know about this. We just tell us is the line offset to the left or right. And here, basically what we do next is we make one of these elements offset into the opposite direction. So this goes to the right and left. Like in the TMS experiment before, we are at 50%. So they cancel each other. And it's a very specific effect, because if we move this outside of the focus of attention, you're back to square one, you ignore it. And we have many, many experiments which clearly show that this is basically a period where you integrate information and it's mandatory. So even if you ask subject to ignore this, so we explain them the whole stimulus, we say, we give you money or whatever, please don't respond to this one, only to the first one, or maybe also to the second one. They cannot do this. They are doomed to integrate. And that basically holds again for an interval of about 400, 450 milliseconds, depending a little bit on the observer. But if this is at, say, 600 milliseconds, then observers can clearly perceive two offsets in the stream. So again, we have this period where we basically integrate information in a mandatory fashion. And I think this all makes sense. Because, for example, if you're watching a car that runs through the night, you know, you have all these whatever reflections and there are whatever background and other cars and so on and so forth. And so if you're interested in the color of the car, each photoreceptor gives you a very noisy estimate. It's not good. But if you average information across the trajectory of the car, you get a much better estimate. And I think that's exactly what's going on in this paradigm I just showed you. It is just a toy version of making sense of a small period of time. And in some way, yeah.
I want to ask, so do you think we have an ongoing temporal window of accumulating information of about, let's say, 400, 500 milliseconds? That is all. I mean, the binding is continuous. It's not that we, not in scientific laboratory experiments, but in daily life, we don't have a sense of jumps or pauses in the integration. And so I'm thinking whether it's some continuous that is all accumulating the information across the last, let's say, 400 or something milliseconds. And maybe we don't need to do a lot. Maybe the system is efficient in continuing the binding of what was already happening unless there's a change. So maybe the issue of static displays in scientific investigations is what actually makes it very difficult for the system to integrate, because that requires a new accumulating window or a new buffer, whatever, while if you are just adding what you have now, maybe the binding or the duration of the buffer is actually shorter.
Okay, that's,
I don't know.
No, very good question. And we of course try to address all of them. I think there's three issues here. The first one is, yes, I believe, you know, with the Team S, we showed it in some way, you know, the stimulus is presented for a very short time, 60 milliseconds, and still people respond, or at least what they perceive occurs much, much later on. You can sometimes respond faster. So that's, I agree with you. I think also under normal conditions, just in everyday life, we basically have these 400 milliseconds. And what we believe is that the unconscious processing during this period has a high capacity. It has very good temporal resolution and very good spatial resolution. That's what we show in some way. But and that's the third point. We believe you are not aware of this. So it's unconscious and you're not conscious during this time, or you have a constant percept. That's the idea. And then you get all the information. You know, like with the car, you don't see what's going on at the trajectory of the car. You just store the entire information along the trajectory. And then at the end, you basically tell, oh, that was the color of the car. And also that was the motion of the car. That's what we believe. I know it's a little bit counterintuitive and most people don't believe it, but that's what our experiments show to some extent. And I think we don't perceive the gaps because simply the consciousness has a low temporal resolution. The unconsciousness is high, but the temporal resolution of the consciousness is quite low in some way, and that's why we don't perceive it. And what we think is basically related to the eye movements. We know it's not the same, it's independent, but what we believe is you make an eye movement, and we know that very little information goes across saccades. And then basically the period starts, you accumulate information, you tell what was going on in the scene, and then you make the next eye movement. And it's potentially a similar mechanism. You also don't see the eye movement and the gaps. That's a little bit the idea. So we believe that there's these iconic memories, the saccades, and these windows of integration that in some way are linked together but not identical.
Michael, can I ask a quick?
Yeah.
Can I ask a question?
Ruti.
Hi, Michael. This is Ruti.
Ruti,
Hi. What do you think about masking? We know that when we have like forward and backward masking, the masking is successful if the SOA is 40 milliseconds. When it's above 50 milliseconds, the masking is not working.
Yeah.
So if there is this integration or accumulation of information that you are talking about that takes about 400 milliseconds, I don't see how masking is working the way it does.
Yeah, but what we would say is basically, but sorry, what you basically measuring with the masking is the unconscious processing. You basically have the mask and it tells you about the detectors. It tells you about the temporal resolution of your low level unconscious, of your detectors working in an unconscious mode. So in some way, if the SOA is short or long, you basically integrate simply by an unconscious mechanism. But the product of the masking itself becomes conscious much later. That's more or less what you know, this is nothing else as a masking paradigm. It is just an, you know, if you take off just take the first and second frame here with this one and this one, it's just metacontrast masking. It's just many lines. But we can extend this. You know, if we add more lines, the masking continues. Actually, it's not a masking, it's an integration. And we also believe that masking is more an integration of information. It's a matter of sense making. Yeah. See, this element here is carefully analyzed by the system. It's here, you can see it, but not where it is. You see it at other elements. So the brain basically just had transformed the stimulus of the experimenter into one which we perceive, which is not the way as, okay, that was very badly expressed. What I want to say is the information is there and it might be expressed at a location where the experimenter doesn't expect it. And I think the same is true in masking. The target information is somewhere. It's just not where we are looking for it.
Can I make a comment?
Yeah.
Yeah, I really like your point of long temporal integration. By the way, we see evidence for it in the activation in high order areas that take hundreds of milliseconds,
Yeah.
even when the stimulus is very short. But I think your metaphor of averaging in the color domain is a bit missing the point, because I think the information is not really integrated in a way in accumulation or averaging, but it is combined. I think you actually said it with the thing. So if you think, for example, about apparent motion,
yeah,
it's also integrating over a long time. You don't see the initial bar, but
yeah.
it is combined. It's not average out. It gives you information about velocity.
I completely agree with you. The integration is a bad term here. And it is not a low-level integration, it's a high-level integration. For example, if I take these lines off here, you don't integrate. It is a period of sense-making where information is integrated in a, how should I say, in a higher cognitive, it's not cognitive, but in a higher processual fashion. It is not about just having—
Maybe combined is a bit better term, yes.
Yeah, and it's not only integrated, it's often also segregated. For example, as I said, if I take these two frames off or just one, then this element becomes visible. Or if, for example, I put an occluder, if I take this off here, this element, this frame, you can see the two separately. Actually, I have demos, but they don't work. But if I then put an occluder here, you get the integration. And actually, you get also integration if they go in the same direction, it gets better. But I completely agree with you. I don't want to say this on a photoreceptor or low level integration. No. What we believe, it's more a Bayesian type of thing. It's really a period of sense making where we come up with the best percept. And I agree with you with the physiology. If you look into the physiology, you see that basically, even for short stimuli, all the information arises in the EEG or also in the monkey, I don't know, around 300, 400 milliseconds, quite late.
It also resembles Shaul Hochstein's ensemble statistics in a way. Okay, I'm sorry.
I think this is, in some way, that's the same thing. Basically, the question is basically what goes together as a texture or as an object and what does not. And the brain solves this. And then it tells us and says, oh, this is basically a background. This is background you should be ignored. And this is part of the foreground. For example, in the crowding, my point of view is, I'm running a little bit out of time, but I come to this maybe in a second later. We are almost done. Okay. So yeah, and again here, so for example, if the lines go in parallel, we can play the same game. You can attend here. The verniers here, they are all straight. Then we add this one. Performance is good here. Then we add this one opposite direction. They cancel. And now we add a line here to the left. And all of a sudden, this basically does not integrate. All of a sudden you see only this element, not this one. And that doesn't integrate. Yeah, it's here again. This is the same element. We just added a line here. But now we think that now you basically, the integration changes and you basically integrate this straight line with this one, but not this any longer. If we move this line to the left, then again, they're in the same stream and they basically integrate. That's basically just what Rafi just mentioned. It's a combination. It's something high level stuff. It depends on the grouping. Yeah, on the low level, everything is the same. It is just this little line here to the left and right that changes things. And we think this is a standard situation. Basically, the information comes in. You first solve all these configuration problems. Yeah, what we just had with the summary statistics in some way. It is just a dynamic version of this. And the brain decides what belongs to each other. So in this case, the feature can go in both streams for whatever type of reason. But if you disambiguate this in some way, if you have two streams in parallel, then these elements integrate and not these ones. And this difference here is just 200 arc seconds. It's really tiny. And the brain is doing an extremely precise job by basically first solving all these figure ground, this grouping, whatever type of problems. And then basically, after this is done, it cannot be otherwise. Then basically the integration takes place. And it depends on very, very subtle differences. And this is quite measurable effects. So what we believe is that we have shape processing across the entire spatial temporal visual field. So you need to know what's going on at one point in the visual field. You need to know what's going on in the entire visual field and across a substantial amount of time, say 400 milliseconds or something like that. So in some way, we cannot reduce vision. We need to understand how these whole configurations work and how basically they create the percept we have. And I think it's really the appearance, the percept itself. That's a little bit what you said, Sharon. How it looks like, that basically determines whether attention can access the stimuli or not. And in some way, you can see this yourself. You know, looks much easier than this one. Even the elements are the same. Yeah. So interference occurs only within spatial temporal groups, but of course we need to compute them first. And we're spending a lot of time finding out right now how this works. And I have to tell you, many have tried and failed, and I think we are on a good track to fail too. Yeah. So what we believe is in some way, all the information is stored here nicely. They interact with each other in whatever type of iconic memory. And then basically it's collapsed and we have a conscious percept. And I don't want to spend time on this. We're trying to model this right now, and we have tried all models that are available. For example, the CNNs, this complicated figure just tells you CNNs can't do this. This is just for the crowding. Sorry, this is just for the crowding here. And what we're doing right now is together with Craig Francis from Purdue, we have huge simulations with millions of neurons and billions of connections. They mimic a little bit the visual cortex. They have LGN, V1 neurons, but they have all these complicated recurrent processing, and they basically are made basically similar to Kosberg's models. They are made basically to explain when elements go together by illusory contours and things like this. And the philosophy, just to shortcut this, is very rather complicated stuff, takes often weeks to run just one stimulus. And the idea is in some way the following. If you have a stimulus like this, usually the brain interprets it in a spatial, temporal, and three-dimensional fashion. So the idea is a little bit, for example, if you have an image or in a real-world scene where there is a fence in front of a house. On the retina, you don't know exactly which lines belong to the house and the fence. But what the brain tries to do it tries basically to segment the stuff. So if you have now an array here of squares, you can group it. And you can potentially send it to the background. And then you can take the Vernier stimulus and you basically put it in the foreground. And that's basically what the model does. It basically segments the visual scene into what we call segmentation layers. They are not super physiological, I know. But in some cases, what happens is, like with the one square condition, they go in one basically segmentation layer. And then you cannot access it. That's what we just talked about with the attention. Then you cannot access it. You basically end up with a texture of type blah, 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 and you have no access to the individual features. But if you have a regular array, which obviously is different from the target, in this case, the Vernier, then you put it in a different segmentation layer, and the Vernier in the other segmentation layer. It's like with the fence in the house. And then it is just in this segmentation layer. It's a little bit like in visual search with the maps. And then you can access it, and then you have very good performance. And this happens, of course, over time. You need to compute what elements belong to each other. And you can basically see then that here the squares end up in this one, and here after, okay, here it's basically 450 milliseconds. You have a good grading here, and the Vernier is a different segmentation layer. That gives you good performance. That's one of the models we're testing right now. Most models can't do this uncrowding, but there are some deep networks like the capsules, they can do this as well too. But they also have an explicit grouping stage, and the inference happens after the grouping. That's the idea. Yeah, that just shows we can do this with the model. We don't have 7 because it's too big to run. Okay, so I'm done. So I hope to convince you that understanding the visual system, just looking at one point in the visual field, doesn't help. To understand what's going on, we need to understand how all elements group together across the entire visual field and over substantial times, and that the entire configuration matter. And potentially we can break down the complexity of vision, but potentially we should not look at locally confined mechanisms, but potentially more at mechanisms that cover the entire visual field. That could be something like this, that maybe the primitives of vision are not lines, maybe the primitives of vision are entire scenes that have certain type of characteristics, but that's pure speculation at the moment. I'd like to thank the Swiss National Science Foundation for very generous funding, and I'd like to thank you for your attention and staying with me over this long time. I think I spent a little bit over. Thank you very much.
You were great. Let's all thank, you can unmute yourself, and let's thank the speaker, Michael Herzog, and everybody is welcome to ask questions. You can unmute yourself and feel free to ask questions. It was a fascinating talk, by the way. I'm still thinking about all the integration aspect, but let others.
Michael, I have a question. So your conclusion is that we cannot just, as basically the early models, that we can decide upon the local information, basically. Local information is not enough.
Yeah, that's exactly the point. Just looking at one point, I think the entire philosophy is in danger. The entire philosophy usually says, oh, let's find out what's going on at one point in the visual field, all over the visual field, and then we combine this. So it's independent. Basically, before you understand what the whole is, you basically understand what the parts are. And from the parts, you can determine the performance on the whole. But that doesn't work. I think this was quite obvious. You know, I showed you the configuration. I can show you more of them. And I can ask you what is predict the performance on the Vernier, and you cannot.
But as far as I remember, you did also backward masking.
Yeah.
That to prevent the late information, basically. Right? So, when you do backward masking, basically, you can basically stop the information over time. So I don't remember what will be your conclusion from that.
Yeah, it's the same thing with the masking. The point is to understand what's going on at one point in time is determined by basically what's going on across the entire, I would say, half segment. I think this is the basic unit of information processing. It is basically the intersaccade time under normal condition, but we can show that they're independent. You get even the integration across an eye movement in this line. But we think that this usually goes together and that this makes the basic unit. And again, with the masking is the same as with the space. To understand what's going on at one point of time, you need to know what was going on before and after, within this period of 400 milliseconds. That's a little bit the idea.
But what I'm asking, what happens if you force the subject to respond immediately, not to wait over time?
They're doomed to integrate. We did this. We say, respond as quickly as you can, try to ignore it, try to escape it. It doesn't work. That's the point. It's a little surprising. I agree with you. It's a little bit surprising if we have this, what we call sequential metacontrast paradigm. If we ask the subject to be as fast as possible, they still take the second information into account, even if it comes quite late, 300-400 milliseconds.
Yeah, but just to comment, there are conditions where you are much, much faster. I mean, the experiment that Torp was doing that showing, you know, you can do face recognition extremely fast. It's not an obligatory step. It's only under certain conditions that you get it.
Yes and no. I completely agree. And also, like if you do sports or things like that. Yeah, but what we believe is, I think it's an important point. We're not saying you cannot act beforehand. You can act beforehand because, you know, as I said, if you do fast sports or the recognition, it depends a little bit also on the stimulus configuration. Of course, if this is just a flash, you can be faster. But I think also Simon and me agree on that. Basically, you can have the object detection first earlier, but also he would say that the consciousness, the full percept arises a little bit later. So you can basically do this task. You can have I'm not saying you can never ever recognize something earlier than this. That's not what I'm saying. I'm just saying the full percept comes later, even if it is short. And if you have information in the stream, then basically you're doomed to integrate. If it is just a flash, you can wait. And I'm sure, I think we tested this to some extent. If you have the faces or the animals he presents in a complex scene, then the reaction times also go up.
Can I ask a question as well?
Yeah, sure.
Thank you. I'm Chagit. I'm from the computer science department, but I have an interest in cognitive psychology. I have two questions. The first, in your example with the vernier and the stars and the squares, I assume, if you can put it on, I assume that there is an effect of how similar the actual target is from the flankers, so that if you take, that image, that one is good, or 39. So if you take the flankers and just rotate them by 90 degrees so that the rows would look more horizontal versus vertical in the background, right? Yeah. And they align less with the target vernier. That would affect the right?
Yeah, it would affect it right away. Yeah. No.
The more similar it is, the stronger the effect, right? The more masking you get. Okay?
Exactly. That's exactly the point. Okay. Even sometimes subtle changes. For example, if we have all the squares in the row, and you just jitter the position of the squares a little bit amongst the y-axis, the effect often goes away. Or, for example, if you fill the squares with a line, then all of a sudden it doesn't work any longer. So indeed,
because it becomes more similar to the target.
Exactly. It is all about if you can segregate the target of the configuration. And what matters is some, computer vision people, I think they have very nice algorithm for this, basically determining to what extent configurations are similar and not. I think this is what we need to think about it. But we horribly failed up to now.
I saw that you said CNNs, right? To try. Did you try RNNs, recurrent neural networks?
Yes.
They seem to be more suitable to searching for patterns and repeated patterns and things like that.
Yeah, we have an entire paper on this, and most of them fail too. Unless you these capsules networks, I don't know if you know them by Geoffrey Hinton, they basically do a very good job.
Okay. Mm.
But this is front-to-end training with the stimuli and things like this. And they basically have these agreement mechanism, which elements go with each other. And then they do a really good job.
Mm. Interesting. Okay, thank you.
It just came out in PLS computational biology.
Okay, I'm going to look for it. Thank you.
My pleasure.
I can I ask a question. I'm sorry I can't put my picture in the moment because of the arrangement of the computer. But the question is, what happened in the case of, you said that this integration process, or it happens in the intersaccadic time, kind of, in a way.
I believe there is a strong link, but we did this paradigm where we basically moved the lines. Oh, I can show this to you. Here. If we basically...
We don't see now.
Yeah, it seems that the PowerPoint has gone away. I don't know.
You need to share again.
Maybe it's the sharing.
Yeah.
Yeah.
Okay.
Yeah.
So if you have this paradigm and you make an eye movement here in between, you still get the integration. So the window of integration is stronger than the saccade. But the big question, it's independent. But the big question is, what starts a series? What starts such a period in real life? Because in the lab, of course, there is a dark screen and something flashes, and okay. And then we have shown that starts a period. If the first flash starts a period, because if you put the element later on and then the integration doesn't shift, the first thing starts a period, that's fine. But what is it in real life? You know, in real life, things change all the times in a busy city. You know, there's millions of signals that could start the sequence. So what we believe, it is usually the saccade under normal conditions that starts a new window in some way. It's a very strong trigger. The entire visual scene changes. And we know that the memory is basically released. You take very little across this. So we think that there is potentially a link, but how it is exactly, we don't know. And
okay. Yeah, I think you answered my question, but yeah, what I wanted to say that there are saccades that happen, like a corrective saccade or microsaccade that happens really fast.
Yeah.
So then might be in the middle of this window, or
they are
in the middle. Okay.
Yeah, they're in the middle of the window, and usually people talk about whether these microsaccades are good or bad. We would say that it doesn't matter too much because you buffer information anyway. So it's in the buffer, and some small positional jitter doesn't matter too much because basically it doesn't matter for the moment. What matters is just the question you ask to this window of integration.
Yeah, because my thought was that if maybe you couldn't make this a proper decision about the information you receive, so you make a small saccade to get more information.
Oh yeah, that could be. Oh, that's a good point. Oh yeah, I see your point. So what you're saying is, if you're not happy with the input at the moment, you basically small saccade, which you basically then can use during this window of integration to improve performance. Yeah, that's a good point.
Yeah, that was my thought.
Yeah, good. I never thought about it. Yeah.
Okay.
Yeah.
Thank you. It was fascinating presentation. Thanks.
Thank you.
Time for another question?
Yeah.
Definitely. I can say if it's okay.
Yes, so I'm puzzled or intrigued actually about the discreteness claim. So I think there's a strong arguments here in your presentation about having a window of integration, but maybe I think Sharon hinted to it in her question before. You could think of a moving window, right? So it's not this, there is a window of integration, but it moves smoothly.
Yeah.
So you can say that your instantaneous conscious percept is influenced by the history of 400 milliseconds or so.
Yeah.
What is the strong argument for the discreteness?
Yeah, okay. Of course, you can have basically a sliding window or a convolution, but with a convolution, you basically end up with too many percepts. So in this case, you would potentially have information, say the window of integration would be somewhere here. So it contains the first vernier. Then you would see at one moment of time, with a sliding window, just this vernier would say, oh, it's there. Then it moves, you integrate, and then you have a third window here, and you would see only the last one. So you in some way would have 3 percepts across time. This one first, the integration, this is not what happens. Also what Rafi said about the apparent motion. It is not that we see something static, moving, static. We just see one motion. And that's basically the argument why we believe it is not, it can't be continuous. Same here.
Could it be task related? So because your task is actually to respond at the end of the sequence to a question, then as the sequence ends and I have to say whether I saw the first line or I didn't see the first line, then I use the last buffer I had of 400 milliseconds, and that's my answer.
Okay, we cannot rule out certain type of Dennett stuff where you have some fleeting consciousness. But we asked subject to basically respond to the first one, respond as quickly, all these type of things. It doesn't work. For example, if I have a vernier here, and I have another one here which is outside the window, they are much closer in time. They don't integrate. They integrate. They are 300 milliseconds away. They are 150 milliseconds away. Actually, they are 450 milliseconds away and they are 150 milliseconds away. They don't integrate, they integrate. It just came out recently where we showed all these type of things. So that basically tells us, yeah, here is a line, here's a break, and there is integration before and after. And as I said before, you don't have this continuous percept because then you would have 3 percepts. That's why we believe it's discrete. But I completely agree, it is highly controversial. And it's very counterintuitive.
But fascinating.
Yeah, if you don't like it, don't worry. Even one of my collaborators, Haluk Ökmen, doesn't like it. So
it's not that I don't like it. Actually, it fascinates me to think that this is the way it is.
Yeah. We have a paper that just came out recently. It's called Dizzy Traudi, and where we have this basically written down. There will be one coming out in TIX2, which basically summarizes. So if you send me your email, I would be extremely happy to send this to you right away.
Thank you.
Can I ask another question, Sean? It's time.
Yeah, no problem. I mean, I'm not everybody. I mean, as long as Michael has time, it's fine.
Time for the entire day. It's not. I have a defense actually of Leila Trizita-Udi at 2.
So the talk is very intriguing, and I'm thinking about. I largely agree that you show evidence that the perceptual outcome of a local element is determined by the whole picture, right? The whole stimulus.
Mm-hmm.
But then I'm not sure I agree that the building blocks of the perception are the whole scene. I mean, we know that the vision starts in local measurements, and in normal vision, you do have the option to focus your attention on a local element. You don't see it, so you don't see it in very briefly. You can move your eyes and you localize, and then what we know about local processing is valid and important.
Yeah, I'm not denying this. I'm not saying, okay, I think we need to distinguish between local processing and perception of local elements. So I completely agree with you that under normal conditions, what we want to do is in some way, not always, sometimes you want to see more the whole, but sometimes you want to focus on something like a detail or something like this. And of course, the brain tries to be able to extract this information, and sometimes it's successful, sometimes it's not. But my point is that the perception of this local element will be always determined by context.
Well, you can isolate a context to some extent by focusing your attention and focus.
Sure, that's what I believe.
Increase the effect of the content, and then the local element have the major.
No, that I agree on. I would say, actually, we do some experiments with attention right now where we basically have these sequences, not this one, but the crowding with and without attention. But the point is, what I believe is basically, it's in the model in some way. Basically here, the question is, of course, if you have the pre-attentive, that's what we believe, or the partly pre-attentive stuff that basically puts in a different segmentation layer, you can access it. But if they get into one segmentation layer, you cannot. That's the point in some way. So I agree with you. Of course, in a normal scene, you have many elements in the segment, in many segmentation layers. And the attention basically just retrieves one of them. That's this change blindness thing. And the rest is gone. But under certain circumstances, you basically are not able to access this. It's like in your MIB, all this information is gone, if you want it or not. I think it's the same thing. I think like in the MIB, you would get similar things like